Yeah, you might want to click, you can click continue. There's 2 people in the waiting room. Jeff, can you check those guys in?
Yep.
We are all sitting on land that was previously inhabited by indigenous people of Northern Illinois. And I want to acknowledge that as we start our presentation. It's something that the Forest Preserve has started. They did a land acknowledgement last fall, and it made my heart sing that recognition was given for this land that was so cared and tended for preciously by original peoples. And in many ways, we're trying to get back to that. And our program that many of you have tuned in today is to help bring our waters back into healthy, vibrant ecosystems that will support a number of different creatures. So in the spirit of recognizing the original people that were here, I welcome you today. Thank you for joining us. This is a program from the River Prairie Group of Sierra Club. I'm the chair for the River Prairie Group. Jeff Garris is our vice chair and has been so helpful in setting up our programs for many years. And I really appreciate him so much for that. I will just give you really little quick elevator speech for Sierra Club, and then we'll have our program. You should know that for Sierra Club in the United States, there are 64 chapters, and a chapter is a bigger entity. Illinois, the boundaries of Illinois are the Illinois chapter. The reason there's more than 50 chapters is California has 7 chapters, and there are some other weird ways of dividing up. But anyway, there are 64 chapters. Now, Illinois is a chapter, and within Illinois, we have 14 very active groups. The River Prairie Group is one of the most active, but they're throughout. There's one named the Shawnee Group in Southern Illinois. There's the Sangamon Valley Group near Springfield. There are a number of different groups that are active in different parts of our state. Let me just tell you a little bit more about the River Prairie Group, because that's the group that's hosting the meeting today. We have over 3,000 members here in DuPage County. Our boundaries are DuPage County, and we have a number of programs that are ongoing. In the best of times, we meet in person and we hug and we greet, and things have changed now because of the pandemic, of course. But we still have an outings group, and we have probably one of the most active outings groups in the state. We have about 16 outings leaders that are continuing to plan a few events. We don't do overnights, and we don't set up carpools like we used to, but there are some hikes. So if you look on the calendar, the events calendar, you can always Google what's happening with us on River Prairie Group Sierra Club, and check the events calendar for any upcoming outings. Outings are a great way to get started with the Sierra Club. John Muir was our founder, and he took Teddy Roosevelt out into the wilds to learn to appreciate the wildlands, and that's what started our national forests and a lot of our open space preservation. So the Sierra Club was founded to help ensure that the government did the things that they intended to do to preserve the open space. So outings is one thing we do. We do a river monitoring program, and Bob Barbieri is the lead on that. And Bob's actually here on the Zoom. Do you want to say a couple comments about the river monitoring program briefly?
Oh, yeah, let me, I'm not, okay.
You're
I thought I was muted, but I'm not. So yeah, we do water testing of the local watershed monthly and quarterly. We expanded to include Salt Creek and the East Branch of the DuPage River quarterly, and then every month we do the West Branch. And we're looking for nutrients and also chlorides and ammonia. And we've been doing this for almost 20 years. Actually, it's over 20 years now. And if you want more detail, we just published a report last year, and that's on the website. So if you go to the RPG website, you can scroll down and And then there's a link to the report. And
Yeah. And
so
Bob
we got a
wrote
good
it
team
in
of volunteers,
stellar.
and we're always out there, rain or shine, freezing or not. So
Our water program is something to be very proud of. And thank you, Bob,
thank you.
for talking about it and for sharing that for so long. We also have a little section of the Forest Preserve of DuPage County in Glen Ellen called Churchill Woods. And Bruce Blake is the steward for that area, and he leads some restoration efforts there. And we don't have them on our website quite yet for the spring, but he will put them on there. And if that's something you want to get out and cut some buckthorn and pull up some honeysuckle bushes, we welcome you to join us. And generally it's on a weekend morning, and meet good people and get a little exercise. And it's a beautiful area. It's a very special remnant that's never been farmed or plowed. So it's a neat area. We also have programs, of course. We have a very active piece of the Sierra Club that is a cooperation with 3 other entities. We have something called the DuPage Monarch Project. And our brand new liaison for that is here, Kathy. And thank you, Kathy. She helps share the word of the DuPage Monarch Project with Warrenville in Bloom, which is a very active gardening group in Warrenville. But anyway, the DuPage Monarch Project is a joint effort between the Forest Preserve District of DuPage, the Conservation Foundation, Wild Ones of Greater DuPage, which is a fabulous native seed collecting and preservation group, and the Sierra Club. And the 4 entities work together to encourage mayors of communities, city governments, and park districts and libraries. Please sign a resolution, and in that resolution, please set forward practices for land that you control to plant native species and plant healthy habitats for monarchs, including milkweed and pollinator species, pollinating species for them to eat, as well as resisting the use of pesticides and things that will be damaging to them. So we have 33 signatories. Some communities have both their park districts and their mayors and city councils on. So it's a very good effort. So that's the DuPage Monarch Project, and you can Google just DuPage Monarch Project and find out a lot more about that. Lastly, we do some work endorsing political candidates. We are nonpartisan. We endorse Republicans or Democrats, but we endorse candidates based on their track records on environmental friendly practices. And after they're endorsed, we work hard to get them elected, and you can be part of that if you want to. And after we work hard to get them elected, every year this Illinois chapter, the statewide Sierra Club, chooses some priority legislation, and we knock on the doors of our state reps and our state senators and say, these are our bills for this year, and we hope you'll help us get them through your House or your Senate and get them across the governor for signature. That is how we got CEJA passed, the Clean Energy Jobs Act, which has a little different name right now, but that's what it is. And that was a 3-year effort, and it is monumental legislation, a model across the United States for how a state can bring clean energy into their into fruition. So yay, that's a real good thing. I've talked long enough. I'm so glad you're here for our program. I guess I could say, does anybody have any, if you didn't realize about a chat, if you want to throw in, Jeff's excited to go biking. I see folks who are excited. Oh, Mary Verg wants to go birding. Listen for those cardinals coming back. They've been here, but listen to them start to sing. Hiking in the forest preserves. Many of us, if you want to throw what you're looking forward to the spring and the warm weather, please feel free to throw that in the chat. And as you do that, is there any quick questions that I can answer for anybody? Okay, let's hit it. I would like to introduce Carrie Ann from the Forest Preserve district of DuPage County. And Carrie Ann is an environmental interpreter for the Forest Preserve. She does educational programs for them, has done a lot of these Zooms and different kinds of electronic teaching. And she especially enjoys freshwater biology, animal behavior, native gardening and design, and all kinds of STEM professional development opportunities as well. So very exciting. She's a certified interpretive guide for the National Association for Interpreters and a master naturalist, which is no small feat to accomplish that. We are so happy that you are giving this program for us. I will turn it over to you, Carrie Ann, and thank you so much.
Awesome. Well, thank you. Let me go ahead and share my screen here. Oh, you know what? It's disabled, so I'm not able to share my screen right now. Jeff, you might have to give me some permissions here. So today I'll just start talking. Today we're going to be talking about aquatic species recovery in DuPage County, and more specifically with that, we're going to be talking about freshwater mussel propagation. Freshwater mussels are something that, or they're organisms that many people don't know exist. They don't know much about them, and I'm a super nerd for them. They're very interesting once you get to learn about them and their biology. Let me see if I can share. Nope. So
no, I'm trying to get to it. People are joining, and so you keep moving all over in here. Can you do it, Jeff? Can you try also? Oh, wait, you're there. You're there. Yeah, you should be able to do it now.
Okay, let me try.
I'm sorry.
No worries. And hey, if any of us have learned anything the last like 2 years, that like nothing ever goes to plan when it comes to technology, right?
Do you want us, if people have questions, and here I am interrupting you now, but would you like me to ask folks to put their questions in the chat, and then we will just mute during your talk? Or
yeah,
how
that's fine. Either one. I don't mind either way. Okay. If you have a burning question that you would like to ask out loud, that's fine. Or if you have a question you just want to put in the chat and I can answer it later, that's fine too. So just to confirm, you all can see my slide, correct? Adjust my slide. Okay, awesome. Well, let's hit it, because I can talk about this for a very long time. So I'm going to try to keep myself to like 30-40 minutes. So let's get going. So overview, what I'm gonna talk about today, we're gonna cover what is a freshwater mussel, the life of a freshwater mussel. So what does that look like? What do they do? Why should I care about them in the first place? And then propagating freshwater mussels, which is the really cool science part of this whole thing. So what is a mussel? A freshwater mussel is related. They're related to clams. They are bivalves, so that means they have two shells that are joined together. They are related to things like oysters, scallops, things like that. Larry, I love that your cat is joining us right now. I can just see him in the video. So they are in the family of pearly mussels, or Unionidae. This is what freshwater mussels look like. Typically, I should say. They all look very different, and they all have very interesting names. So the outside of the shell is hard, like I said. The thing that I wanted to point out on this slide is right in the very top of the dorsal view. So on the top is the beak, or also called the umbo. And that's a very important part of a freshwater mussel, because that is how we actually, or that's like one of the main things that we use to identify them. When you get into the field, or if you are walking in a river and you start to see freshwater mussels, they kind of look the same. They kind of all look the same. Some of them are very easy to tell apart. Some of them are very hard to tell apart. So the umbo, or the beak, is the spot on the shells where they sort of connect. And so that's one of the important parts. The other thing is lateral teeth, or the pseudocardinal teeth, which is on the bottom picture there on the right. Those are a little trickier because you kind of have to see inside the mussel, but for now, we're just going to be talking about the umbo. What does the inside of a freshwater mussel look like? The inside doesn't look super impressive when you just look at it, but this is where all the stuff is, right? On the inside, we have the hard shell on the outside and all the stuff on the inside. So what I like to do, and especially if I give this presentation to students, I like to ask, look at the inside and look at that diagram that's labeled. what are two things that stick out to you that are not there? And you can certainly answer out loud during this part. So what are two things right off the bat that you notice that freshwater mussels do not have? Larry's pointing at his eyes. They do not have eyes. So that is one of the two.
Mouth?
They
Okay.
do siphon in water and then push out water. So I think that in this case, the in-current siphon would be considered a mouth. The other thing, the eyes is one of them. The other thing is brain. Yeah, so they don't have any sort of organized brain, but they are alive and they are an animal. So keep that in your brain as we continue on through this presentation. Do female and male mussels, do they look the same or can you tell them apart? Short answer, sometimes. Some species look different and you cannot tell them apart. Other species, they do look a little bit different. So in those sexually dimorphic species, or the species that you can tell apart, the females, the way that I describe it is very scientific. I say they look poofy. So they look taller and a little shorter on the sides. The males are more elongated. And the reason those females kind of, to me, look poofier and shorter or truncated is because they are going to be holding those eggs, or they're going to be holding the young. So they need more space to do that. So instead of becoming longer like the males can, they have to be sort of truncated and taller or poofier. Where do mussels live? Mussels generally are living half buried in gravel, sand, or mud on the bottoms of streams and lakes. In DuPage County, we do have mussels in our lakes, but most of them, I would say, are found in our streams or our rivers. The spot, believe it or not, that I've seen the most diversity of mussels, so the most different types of mussels in one spot, is actually the Naperville River Walk. If you walk along there, I've seen quite a few different mussel species just taking a walk down there. So that's a spot that I feel like most people wouldn't think that would have a lot of mussel species there, and Naperville does. So most of the time, these guys or gals are going to be at the bottom of our lakes or bottom of our rivers, and they're going to be half buried. I was fortunate enough to do some mussel surveys a few summers ago and work at the Urban Stream Research Center, which I'm going to talk about towards the end of this presentation. And when you do surveys, you're literally on your hands and knees digging through rock and digging them up on the bottom of the river. So that's usually where they are. And again, this differs by species. So some species really like to bury down, and other species, they like sunbathe almost. They'll just like lay on top of the bottom of the river, and they're like out, just hanging out for everybody to see. So it really depends on who you're looking for, but generally speaking, they're all on the bottom. So kind of a why should we care? Why are we talking about freshwater mussels so much, or why does the Forest Preserve care about them so much? Freshwater mussels are actually the most imperiled animal in North America and in the world. So they are in grave danger of extinction. I believe over 70% of freshwater mussel species that were once around are now extinct or gone. Yeah, and so really to me, they're kind of an animal that people forget about, right? They live on the bottom of our lakes and streams, and people don't see them that much. So it's kind of out of sight, out of mind. So what kind of kicked off this extinction, or what happened? The thing that we like to talk about first, we'll go over a little bit of a brief history lesson. So in 1889, the use of freshwater mussels as buttons was established. If you think about it, when was plastic invented? Not till like, I think the 20s, 30s, 40s. But people needed buttons for their clothes. So what they used was what they had around them, or what they found around them.
It's buttons.
And freshwater mussels were used as buttons. Yeah.
It's crazy.
I know. Isn't it crazy? And by 1920, the freshwater mussel fishery was the largest and most profitable inland fishery in the U.S. So when we talk about fisheries, we're talking about, things that make profits. So a lot of times I think people think about ocean fish or, fish on Lake Michigan and things like that. But in 1920, harvesting freshwater mussels and making buttons from them was actually the largest fishery in the US, which completely blows my mind. Millions upon millions of mussels were used. And there were over 60 button factories located in the Mississippi River Valley. I actually went to school at Western Illinois University, and I would find, I'll show you what they look like in the upcoming slides, some of the evidence that there was a button factory there. And I didn't think anything of it. I didn't really know what I was looking at. When I asked what it was, somebody was like, oh, that's just from the buttons. And I was like, oh, okay. But I mean, they're everywhere. That was near Keokuk, Iowa. I can't remember where the biggest one was. Jesse would remember. The gentleman in this picture here was actually the person who invented or figured out that you could make buttons out of freshwater mussels. And I don't know if it's karma or whatever, you believe in, but he actually died after he cut his foot walking in a river. He cut his foot on a mussel. So he actually died from sepsis from stepping on a mussel after creating this giant fishery. So I don't know, whatever you believe in, that's some sort of justice there for the mussels, right? In my opinion. So what would they do? How would they do this? Harvesting mussels from the river bottoms, they did not discriminate. Anything they could grab, they would grab. How they would do it is they would take, or I should say some of the ways they would do it is they would take boats, and they had these giant, almost like rakes that they would just throw off the back of the boats, and they would scrape the bottom of the river and haul up thousands of mussels. Endangered wasn't a thing back then, or imperiled or anything like that. So they were indiscriminate. They took whatever they could. These are just pictures. This top left is a picture of them just gathering the mussels. Of course, in order to make these buttons, they have to kill the mussels. Anytime you find a freshwater mussel that's alive, and as soon as you pull it open and break those two shells apart, you've killed the animal. So in order to make these buttons, they have to kill the animal. They would steam them open. So they would kind of cook the mussels to steam them open and get the flesh out of them. And this is that kind of evidence that I was talking about that I saw near Keokuk, Iowa. We call them button blanks. So they're the shells that they use to stamp buttons out of. These pictures really remind me of the pictures that we see from out west of all the bison skulls, like the people standing on top of the bison skulls. But this is what they did more locally around the Mississippi. And these are the shells from all the mussels that were harvested to make buttons from. So really the kickoff of their decline, and what, we say is we did this essentially, right? But the button industry is really what kicked off freshwater mussels into their kind of downward spiral. So what else kind of contributes to them being the most imperiled or most endangered animals right now? Besides the fact that we really put a dent in their populations with making buttons from their shells. Dams and impoundments in our local rivers, channelizing and dredging our waterways, siltation from erosion, water pollution, loss of in-stream habitat, loss of fish hosts. We'll talk a little bit about that later. Commercial harvesting again, so for making buttons, pearls is an industry too that freshwater mussels have certainly taken a hit from. If you want me to talk more about pearls later, I certainly can. Just put it in the chat and we can talk. I didn't want to go too far off tangent today. And then of course, introduce native species. So all of these things together have created this perfect storm for our freshwater mussels to begin declining. So now I'm going to shift to kind of how these creatures live, what their life history is like, and their sort of reproductive strategies, because to me, this is some of the coolest stuff that I've seen in nature. And I get really excited to share it with everybody. So the life cycle of a freshwater mussel, I like to ask people this too. Have you ever thought about what their life cycle is? Like, not where do they come from, but what does it look like? Do they come from eggs, or are they just baby mussels that grow up into adult mussels? Like, have you ever thought about what that looks like? And freshwater mussels are actually a parasite. They have to be a parasite for a certain part of their life cycle in order to continue on to the rest of their life cycle. So in essence, in order to grow up, they have to be a host. So we call that they're an obligate parasite for a small part of their life. In this middle picture, these are like really zoomed in microscopic baby mussels. Baby mussels, another word for them is called Glucidia. And those Glucidia have to live on a fish host for a short part of their life cycle. It's really only a couple weeks. But what they do is they clamp on to the gills or the fins of a fish. And like I said, days to weeks later, so really only a couple of weeks, those babies or those Glucidia will actually metamorphose into adults or into juveniles, first, and then they drop off into the river substrate. So this is where I would like you to think back about that slide we were looking at that freshwater mussels don't have. So they don't have eyes and they don't have a brain. But in order to get there, if we are female mussels and we have Glucidia that we need to get onto a fish, how are we going to do that without eyes? And how are we going to do that without an organized brain? This is where stuff gets cool, in my opinion. This is where nature gets really cool. Let me check my time so I don't go too far off here. The answer is lures, conglutants, and fish snappers. Female freshwater mussels have evolved an amazing variety of strategies to attract a fish host for their babies to clamp onto and then continue their life cycle. They use lures or these conglutants to attract fish to them. Once the fish bites down, they release their babies, and then those Glucidia or those babies will clamp onto the fish gills or fins, and then those babies swim away with that fish, and they continue on their life cycle, and they are on their way. So this first picture up on the top left is an example of a lure. And what does it kind of look like to you? I know it's not like moving, it's just holding still, but does it resemble anything to you? So this is called the mantle, and that's the part that's the lure. And then up here, this like poofier part, that's actually her gills that are filled with her babies inside of them. So they're swollen because she's got those babies that she's ready to release. So what does this look like to anybody? This is kind of an eyeball. This is a stripe. Here's some fins. Does it look like
we're not seeing where you're pointing.
Oh, okay. Well, then that explains it.
That's why we're being silent.
That's okay. So that top left one is a plain pocketbook mussel, and she has a lure that actually looks like a fish. So she is trying to attract a larger fish that would eat a smaller fish like a minnow in order to release her babies and continue on, so those babies can continue on their life cycle. I have a video. Let's see if it'll play here. I think I have to exit out of a different mode here. Let me know if you can hear, because sometimes I run into issues with people hearing the video. No? Okay. I'll just narrate for you then. This is a great video of an example of what those lures look like. So this is the first one was a plain pocketbook. And these females, look at the color change. She's making that color change on this lure right now. They'll also kind of vibrate it or flick it to make it look like it's a fish that's swimming to attract a fish to her. That fish would try to eat that minnow in quotes, and then she's able to release her babies to continue their life cycle. So this video too, she's like flicking it, trying to make it look like a minnow.
What do you do for a living?
Pause.
Organization.
Okay, here's another video. You may not, can you hear that sound or no? No, okay, it's okay. I'm gonna fast forward it a little bit because I just want to get to the part where you can see the lure. And there's the fish. So here's the fish this is a largemouth bass. And in order for the female mussel to release her babies, she needs to attract that bass to her. And that bass needs to bite her lure. Sorry, I'm turning down my volume a little bit. So I'm just gonna let it play. I know it's silent and it's kind of weird, but here she's gonna start to look at that lure. How much of a, does that not look like a fish?
Yeah.
That she's flicking like that? So these female mussels don't have eyes, like I keep saying, and they don't have a brain, but they've evolved to look like a minnow. She's starting to flick it a little bit faster because she can sense that a fish is near her. How, we don't know, because again, she doesn't have eyes. So in a second here, you're going to see the bass is going to go after the lure. And you'll get to kind of see those. There we go.
Mm-hmm.
So that poof was actually her babies, or Glucidia, that she released onto that fish, so those babies can continue their life cycle. Isn't that amazing? There's so many different kind of lures too. I mean, that's only the tip of the iceberg. You can take a deep dive on the internet if you Google freshwater mussel lures. Some of them look like crabs. Some of them look like insect larvae that smaller fish are attracting. So that's kind of the first part that I think is amazing, is that they have evolved these lures to look like fish. The second half of that is some of these mussels have to attract a certain type of fish only. So some mussels are generalists and they can use all kinds of different species to have their babies continue on their way. And other mussels will only use one species. So they have to figure out a way to recognize that certain species that they need to can have their babies survive. To me, that's amazing that they do this. So moving on, I could talk about this for hours. Why should I care about freshwater mussels? They're in the river. They're in the bottom of the river. We never see them. We don't eat them like we do the ocean mussels, typically speaking. So why should I care that they're so endangered? Well, as I'm sure we're all aware, especially people in this group, we never know what we have lost until it's gone, type of a thing. And everybody and everything has their role. Freshwater mussels provide a ton of ecological value. They provide food for a bunch of different animals. They provide ecosystem services. They stabilize the bottom of the river. And provide critical habitat to a wide variety of different aquatic organisms. But if there's one thing that I would like for you all to get out of this presentation tonight, why should I care about freshwater mussels? And that is because they are the livers of the rivers. So they are filtering, don't you love that? They are filtering our water systems for us. So, that's the biggest contribution I think that they give for us as humans to care about them. Whoops, I didn't mean to go so fast. So livers of the rivers. And another thing is they're actually biological indicators of water quality. They're pretty sensitive to environmental changes. So we can look to them to see sort of how our rivers doing? Some of them can live in any type of water. Others have more, have like stricter requirements as far as water quality. So you can kind of judge, just like we can use that biotic index for invertebrates. Sometimes you can look at mussels the same way. I threw these pictures in here because we get asked a lot, do mussels move? And you can see from the picture on the left, they do indeed move. They don't run by any means, but they have feet that they use to kind of bury themselves down into the substrate. And that's what the picture on the right is too. There's three different drawings of mussel species in there. And that foot is how they use what they use to like anchor themselves and really stabilize that riverbank as well. Now we're going to get into a little bit of the propagating for conservation. Why are we doing it? Really, when it comes down to it, it's all about biology. The Forest Preserve with several other conservation agencies, we've had the opportunity to restore and naturalize beds and banks of DuPage County waterways. This slide says over 15 miles across 3 waterways. I think it's even more now because it seems like every year we're restoring more and more water in DuPage County. We've removed plenty of dams along the way. And then we're just creating that diversity and preferred habitat for different types of flora and fauna. What else are we doing when we restore a river? We're really, I'm sure all of you are well aware of this, but we're reconnecting floodplain, giving the river what it needs, creating that in-stream habitat, and decreasing erosion. So it's not only about putting species back into the rivers. The first step is restoring those rivers. So that's what we do first. We would never want to put species into a waterway or a water system that isn't healthy for them or that couldn't support them. So really all of this ties together. First, number one, restore the river. And then eventually when we get to a good place, we can put some of those animals or those freshwater mussels back into a river. I like to put these pictures in here just because it does show some of the river restoration that we've done and what a healthy river or stream should look like. This is from Springbrook Creek Phase 2, which has taken place in Blackwell over the last few, I think it's now finally complete, over the last few summers. So it went from being sort of a wetland, marshy sort of habitat that tended to flood quite a bit, and we re-meandered it and restored it into, what I like to tell the students that we talk about river restoration with is that a healthy river or stream should look like a snake, like a windy snake. So you can see in the picture on the left, that's what we went with, or that's what we shot for. And that's why the river looks like this now. So what are our goals for propagating these animals for restoration and for conservation? We're not trying to bring back, so when we propagate freshwater mussels and we are propagating them to put them back into rivers, we're not propagating to bring extinct species back. That's not our goal. Really what we're trying to do is keep those common species common in an urban stream system. We're trying to augment and restore the native mussel assemblage community and the ecosystem health and services they provide. So again, we're really just trying to help restore what used to be there, not so much extinct, but maybe help some of those species that their numbers are pretty low because of water quality or because the habitat was destroyed at some point. And then ultimately, we want those wild populations to be self-sustaining. So we're really just kind of trying to supplement those wild populations until they can breed themselves over time. Like the goal of restoration and the goal of raising these animals isn't for us to do it forever, right? The goal is for us to be able to do it enough to get those species to a place where they can take over and continue to thrive on their own. So again, why are we propagating freshwater mussels? Low population numbers, low recruitment, meaning they're not breeding like they should be, And then low diversity. Historically, there were 16 mussel species known to occur in the West Branch of the DuPage River. 9 have been documented to still be there above the dams on the West Branch. So we went from 16, there used to be 16 in that river, and now we've only found 9, and those 9 are listed kind of right in the middle there next to the picture. So are we bringing back those extinct ones that we haven't seen for years and years? No. Part of the reason for that is that those fish hosts have to be there in order for those mussels to be successful. So it's not only about restoring the habitat, putting the mussels back, but as we learned, mussels need fish. So if the fish aren't there, the mussels aren't going to do well. So there's just so many things that go into it, that it's not just a simple, let's put some extra mussels in the river and they'll do great and they'll help, filter water better because there's more of them. Fast forward to 2012, the Forest Preserve District opened our Urban Stream Research Center. So this is like where all the magic happens. This is where the propagation is actually happening. Like I said, it was opened in 2012. It's a really cool facility if you've never been inside of it. It is generally closed to the public, but we do have public open house days. I think they're in the fall. We did them in the fall last year, and I believe they're on the docket again for this coming fall. So if you have never been in the building, I highly recommend going through. It's really cool just to see a facility like this that was built from the ground up, sort of tailored to propagating freshwater mussels. It's such a new science and such a cool new thing that we are one of only 20 to 25 facilities in the United States that does this, and we are the only facility in Illinois that does this. So it's pretty neat stuff that's happening in this building. So the next slide, we're going to see if this is going to work again because I'm not sure, is a video again. Let me try it a different way. See if it'll do it for me, or see if you can hear. Can you hear that or no? Ah, this is such a good video. Okay. Let me try something else. Can you hear it now?
No, it's Jesse.
That's okay. I can send out links to these videos. Like I can send them to either Connie or Jeff. And if you want to share them or I can stick them in the chat somehow. And then if you all would like to watch these videos, you can watch them, on your own with sound. I don't want to play this one because this is like a really neat video that is really nicely done. So I don't want to ruin it by like talking over everything that they're doing. We'll skip it. I have everything that I was going to show in that video in slides, but I was just hoping that you guys wouldn't have to hear me talk as much as now you have to. So we're going to go through step by step sort of how propagation works and what we mean by propagating freshwater mussels. So step 1 in the process is we have to find a female that's pregnant. So we have to find a gravid female. We then harvest her Glucidia. That picture I know looks really scary because it looks like it's a needle. We actually don't pierce anything in the mussel. It doesn't hurt her at all. We're really just like extracting or flushing out that sac or those gills where she keeps the Glucidia. This is a picture of a plain pocketbook mussel. So we're harvesting her Glucidia. Then next, again, fish is a necessary piece of the puzzle. So in order to get these baby mussels or Glucidia onto the fish, it's a very scientific process of putting the fish in a barrel of water and then adding the Glucidia into the barrel of water and then stirring them together for about 45 minutes. So it's really a simple process, but it is a necessary part of the process because these baby mussels have to live on these fish for a certain amount of time. So stirring the fish together, there's a picture of it kind of right there, stirring the fish together with these baby mussels. We're then inoculating the fish host, and that's a really great picture of the baby mussels on the fish gills, so you can actually see them kind of clamped on to the gills. Next, we have to collect all those juvenile mussels that are now metamorphosed, because that's what they do when they're on the gills. They are metamorphosing, and they are ready to continue on their life cycle. This is a little video of you can see the little microscopic baby mussels. They look just like adult mussels, but tinier. You can see their little feet that are kind of like sticking out. They almost look like tongues, we tell people, but they're not tongues. But that's what they use to like move around when they're little. And this sort of stage of the process is tough, because we have to count all the baby mussels that are dropping off those fish hosts. So after those mussels are ready to drop off, they start to drop off. It's a bell curve, right? So like a few will start to drop off, and then it'll be like big time dropping off. So we have to collect and count all those mussels, and then it'll kind of slow down again. Now, one single female mussel can release 100,000 babies. So we are hand counting some days 50,000 mussels in a day with a microscope. So it's quite a laborious process, but it's important so we can make sure that all of those babies are thriving and that they are all getting what they need. So we count them at first when they fall off the fish, and then we count them again weekly after that. So weekly, these guys and gals are counting 50,000, 70,000 mussels every week. Once they drop off, they go into a rearing system. These are just pictures of the different types of rearing systems. All that it really comes down to is different species of mussels need different things. So that's what all these different systems are for. Once they've grown up in the lab and they reach a certain size, our goal is to get them outside as quickly as possible, not to release them as quickly as possible. But there is something about being in a wild water system or being in a pond that humans, no matter how well our lab is built, we cannot give them everything that they need. So we will get them to a certain point in the lab to where they're big enough to go outside, and then we let them continue growing outside. I think I have some pictures that will show that. So here's just some pictures of growth in the lab. So these babies have not been outside yet. You can see 1 millimeter, 5 millimeters, 10 millimeters. Maybe the size of your fingernails are smaller. And then here's some pictures of, you can show the difference in the growth. So these mussels went into the pond system. So we put them in that wild water when they were 3 millimeters. 1 month later, they were 10 millimeters, just after 1 month. Those same mussels that went into the pond at 3 millimeters were pulled out of the wild water pond system at 4 months, and they were 35 millimeters. So there's some sort of exponential growth that happens when these mussels are actually released into some wild water, which is for us like a very important piece of the puzzle too. Fast forward, all the mussels we'll say have been in the lab. They've grown to a certain point where they've gotten into the wild water, so they've gotten into our pond system. They spent a couple months in the pond. We pulled them, and now they're ready to be tagged. We put 3 different types of tags on our mussels. The 3 different types just mean that we collect 3 different types of data on them. The picture that has the 3 mussels on it, on the right side, that is a pit tag, or like a microchip like you have in your dog or cat. That type of tag, we are tracking that individual mussel as long as we can find it. So we are going to measure it, weigh it, whatever sort of information we want to get from it. The middle tag, the yellow one, is called a hull print tag. That is a unique number that is attached to that mussel. Same thing, we're going to be collecting data specific to that mussel. So both the yellow one and the microchip pit tag, those numbers are unique to that mussel and only that mussel. Then on the far left, the green tag, possibly my favorite, we just glitter these mussels. So we bedazzle them. We glue some glitter onto them, and those are really just absent present. So if we find those mussels, we know, hey, that was one of ours because it's green and has glitter, but we're not necessarily collecting data on that individual mussel. It's really just, is it here? Is it not here? We get these guys and gals ready for release. At this stage, we don't know if they're guys or gals, so we'll call them they. They are put into a trough system where we can kind of lower their temperature or raise their temperature to match what's going on in the river right now. So we get them ready. We put them into bags in batches, depending on the release site and how many we're going to put at each release site. Here's some pictures of us releasing them or where we put them, or like what we do when we release them. If you look at the top right picture, the biggest one, you can look through the water and you can see some orange flags on the bottom of the river, and that's our release site. So we staked out all of these sites, and we keep those flags in the water so we can continually find them year after year to do surveys if we need to. But we will release anywhere from 1,000 to 5,000 mussels at each of these release sites. Here's some more pictures of us putting out some mussels and or finding them again. So those surveys that we're doing to kind of track our progress, or track their progress, I should say. And here's just some survey number examples. So in 2020, a survey was done 3 years after release. We call them dig 'em up surveys. Those surveys that I was talking about where you're on your hands and knees. There are 6 sites that the total release was 5,000 at these sites. And so 3 years after they released, we found about 800 of the 5,000. Does that mean, the 4,000 other ones aren't there anymore or they died? No, that just means that we did not find them within that marked area with the orange tape. So, not bad for 3 years being in a river and having to deal with flooding conditions and whatever else. We did pretty good. After 3 years, we have, or even 4 years now, we have seen females that are gravid or that are pregnant. So hopefully that means that our mission is working, right? Or like our mission is slowly being achieved. These mussels are starting to reproduce, and hopefully they will begin to create their own self-sustaining populations. I'm going to fly through these next couple slides here because a lot of them are just numbers and pictures of mussels. So this is just a list of the species that we have successfully propagated. Because we are the only facility in Illinois that does this, and because we have become so successful, I like to jokingly say we wheel and deal mussels now, but we will propagate mussels for other conservation agencies. So we have propagated mussels for Kane County watersheds. We've propagated mussels to be put into the Chicago River watersheds. So these agencies do approach us to partner now and propagate mussels for them if we can. So we're doing both propagation work and conservation work for not only DuPage County rivers, but also other counties surrounding us as well. Here's just some pictures and numbers, like I said, of some of the mussels that we have successfully reared and released. I have sort of a summary at the end. If you take a look at the names, they have very interesting names. This is just, again, the tip of the iceberg. Some of the names of these mussels are awesome. Creeper is one of my favorites. That's a really neat looking mussel when you see it in person. Creek heel splitter, giant floater, great names also. And here we are at the end. So currently, since we found success in propagating, It took us a couple years. So we opened USRC in 2012. We didn't really gain any major headway or become really successful in propagating until 2016, 2017. And that's just because it's science, right? You learn stuff along the way. Not everything's perfect. It's trial and error, and you learn from your mistakes. In 2017, they were the most successful and were able to propagate and release over 23,000 mussels. To date, we've released over 25,000 mussels into DuPage County rivers and other local watersheds. So currently in the lab, they do have quite a few different species going on. These mussels we're going to be holding in the lab until it starts to warm up a little bit. Then once May hits, this group of mussels are going to go into those pond systems and go into the wild water, and then eventually be released then in the fall. So, sorry I flew through that last part. I wanted to get it all in. Any questions for me? You can say them out loud, or I can just go look in the chat, whatever you prefer.
What kind of spread have you seen? What kind of migration?
That's hard to track. That's something that IDNR has asked us quite a bit too, that nobody really knows how far freshwater mussels move. We're pretty limited. We're really just looking for the mussels that we released around our release site. We're not tracking them, even with those microchips and those pit tags, we're not tracking them super far. And rivers flood. So those mussels can, if they loosen themselves from the substrate, they can fly miles downriver. So that's something that IDNR and other scientists are wondering as well, and we don't really have a great answer to it quite yet. Good question.
Is there any possibility they make it past any dams?
Anything's possible when a river's flooding. But I would say it's not very probable that they're going to be making it past dams. That's a good question.
So Carrie Ann, I think it's amazing that we released 25,000 of these mussels last year, or whatever the time period was. Is the lab at the research center, is it at full capacity now, or because it looks like it's very severe.
I know. Yeah, there's a lot of stuff going on in there right now, but they actually are they just did another round, because I'm trying to meet with the ecologist over there to talk about a different project, and he said we're flushed with juvenile mussels right now, and they're all dropping off. So they're busy counting baby mussels almost 8 hours a day for the next 2 weeks. He was like, "Can we talk in March?" So I don't know if they're not quite at capacity. They're like burning the midnight oil and getting as many mussels propagated as they possibly can. So
I have two questions.
I saw a statistic that an adult freshwater mussel can filter anywhere from like 6 to 10 gallons of water a day. So if you multiply that by 25,000 mussels that were released into the rivers, that's pretty good. Those are some pretty good livers filtering the river.
When they take the dam out of the at Fullerberg Woods, will that then become a watershed where you might place mussels?
It could. It certainly could, because it's within our own watershed. So we'll have to see. It just depends. I'm sure surveys will be done to see what mussels are there and what mussels need to be moved, because that's the other part of any sort of dam removal or river restoration, is moving, like physically moving the animals that are going to be impacted. The pictures of that Springbrook restoration, I was fortunate enough to be working at the Urban Stream Research Center that summer, and we actually moved we moved fish, frogs, turtles, mussels, any animals that we found from the old channel once it was like cut off to the new channel that's there now. So that's a part of any sort of project, is making sure that we're moving any of the animals that could be impacted.
Are mussels, clams, and oysters all the same thing?
That's a tough question because technically freshwater mussels are like freshwater clams. Oysters are a little bit different. I mean, they're all
They're
related.
ocean only?
Right. They're all related. You could say they're like cousins or second cousins. But technically freshwater mussels are freshwater clams. Yeah.
As a kid, I remember that we would always, the oval shaped ones we
Yeah.
referred to as mussels. But the
Yes.
more rounded shapes, we always referred to them as clams. But they're
Yeah.
really,
And
the names now seem to all reflect mussel names.
right. The rounded ones though, and this is where things get tricky because we do have an invasive clam in our waterways that is symmetrical and more rounded, the Asian clam. So that's why I try to stick with personally mussel. So we're not confusing them with the invasive clam if you happen to see one, because freshwater mussels are asymmetrical. If you draw a line down the middle, they're not exactly the same on both sides. Like they're not mirror images, but if you do that for an Asian clam, they are. So that's just my lingo to try to keep things as clear as possible.
And these Asian clams, these are like the zebra mussels?
They are everywhere. They're
Oh, even more
they have
invasive.
well, no, I mean they've been around. They have different reproductive strategies than freshwater mussels do. So they're just generally like more successful. If you walk at Fullersburg and walk along Salt Creek, you'll see them. You'll see the shells because raccoons and mink love to eat those Asian clams. So you'll see their shells. You'll see literal piles of their shells from animals eating them. So
I assume that there's enough host fish or different types of host fish, and that was studied before this project began?
yes. So anytime there's any talk of propagation or river restoration or dam removal, surveys are done. So we know kind of what's there. And that's another reason why we're not putting uncommon or extinct species back into the waters, because if their fish hosts aren't there, they're not going to survive. So we wouldn't just put animals back into a waterway that they're not going to be able to reproduce. So yeah, absolutely. That's a super important part of the project, is like making sure that the correct fish are there before we do any of it. Great question.
There certainly are still wild mussels out there. I don't know how many, but I live on Flag Creek, and the muskrats and the raccoons love the mussels.
Yeah, you can find them a lot of places. It just depends on the quality of the water is like dependent on the what types of mussels you're going to see, I guess. But
So is that becoming a problem with the ubiquitous nature of raccoons these days? Making it, I mean, I think that's an issue for the Blandings turtle. You know, we have carnivores eating them up when we're trying to replenish the stock.
Yeah, I would joke with the ecologists at the OSRC and say, like, I hope raccoons don't find your release sites, and they have not seen that. I think there's plenty of Asian clams and other things for them to be eating. But
good,
because Asian clams are everywhere. I mean, they're just laying all over the bottom of the creeks and stuff. So I think they have other things to keep them occupied. I'm sure they do eat some, but there's not as many just hanging out like Asian clams are.
So the Asian clams are the most plentiful of any of them?
I think so. Yeah. They're not very big either. They're like
the little
maybe like the quarter size.
Size. Yeah, they're quarter size.
Yeah, they're not very big. So their strategy is like, there's a lot of them, you know. Any other questions I can answer? I can certainly look in the chat too.
And the zebra mussels, the big problem with them is that they clog up things, or is it that are they the ones that overfilter the water, or is
all
it
of the above? Yeah. And they can survive. So their babies are almost invisible. They're like super, super microscopic, and they can just be suspended in the water. So they, that's why it's so important for people to drain their boats or drain your kayaks and not transport or transfer water from body to body, because those babies can just be floating in the water column and then you're spreading them all over the place. Whereas freshwater mussels, they are a little more substantial in that they clamp onto fish. So their life cycles are different. And then zebra mussels also can live outside of water for up to 5 days. So that's another thing, is that you have to make sure you're like drying all of your stuff. They just got so much stuff going on that makes them, successful, that it's hard to control them.
So just want to mention that we lost Connie because there's a power outage at her house.
No.
Yeah, like 1,800 people without power down there.
Oh no.
Yeah.
Wow. Yeah,
So
I
have
can
a-
send along
yeah.
links to those videos too. So I will send
Yeah,
them
definitely.
to you, Jeff, and Connie. And then if people are interested, the video of our ecologist from the USRC is a great video. It's a great overview
Okay.
of the process, and a lot of great pictures and videos of things, probably way, explained
Yeah.
way better than I just power explained through that the last
That's
10
great.
minutes.
What should we search for on YouTube if we want to? Could we find those videos? Sorry, my power's out here
Yeah.
too. I'm using a flashlight and a
Oh,
phone.
I can hear it starting to rain too, or rain pretty hard. DuPage Forest, I believe, is our YouTube channel, and you can find the USRC videos on that YouTube page. The other ones, I don't remember the titles of them. I can actually go back really quick and look what they are. Sorry, you're gonna watch my screen flip back.
But we could, we have our own YouTube channel too, so we could put this video there. And then I could send an email out to all the people who registered so they could see a link to this and the link to what you have, these
Awesome.
videos. So we can cover that.
Yeah, that'd be great. This one is titled, let me
Thank
see,
you.
sometimes it'll show me. Natural fish lure, Lampsilis mussel and bass. I can send links too if you don't want to search the titles. And this one is Lampsilis mussels mimicking minnows.
Great. So Carrie Ann, I definitely appreciate this. I think we
Sure.
enjoyed a wonderful presentation tonight. And,
Thanks. Yeah.
and
I
there's just
encourage
so
you
much-
all to come to
yeah.
the USRC if you can for an open house.
Yes.
It's really awesome to see it in person and
Yes.
talk to the people that do the research as well.
Yeah, we'll have to keep an eye out on that for our calendars. I did want to mention something before people start signing off. And we do have another very interesting program coming up on March 16th. It's called Bicycling with Butterflies by author Sarah Dykman. That's Dykman. She's got a book out. She was cycling all over the country, even down into Mexico, and walking to follow the migration of the monarch. And it's going to be an amazing presentation on March 16th. There'll be an announcement for that coming out with our newsletter at the end of the month. So keep an eye out for that. Her book will be on sale, is on sale at Amazon, 50% off. So there's a bargain there. So I'll put that announcement in the email that goes out for this group.
FYI,
Yes.
I'm sorry, I just
Sure.
finished a great book called The Language of Butterflies, Wendy Williams, and it was really good book.
Excellent. So we're all back. I can see everybody now.
Yeah, I was like, I don't need to keep sharing my screen. Sorry.
But thanks, Carrie Ann. I could probably,
Carrie Ann.
if we're done with questions, I can end the recording. But
Okay.
any more questions or anything? But certainly, we want to thank you for your time tonight.
thank
and this
you.
excellent presentation. I think the
Thanks
science
so
that's
much.
going on in DuPage County Forest Preserve is just amazing. So
Thank you.